Hello, everybody, and welcome to this pre-recorded presentation of Tranquis Therapeutics for the Bio, CEO and Investor Conference being held in February of 2022. My name is Sanjay Kekar, and it is a pleasure to be with you here today. I'll begin with a short overview of the company and briefly describe our strategy before covering our scientific approach, as well as sharing some of the in vitro and in vivo data in support of our platform targeting immunometabolic pathways. I'll conclude with an overview of our pipeline and near-term milestones. Tranquis Therapeutics is discovering and developing innovative medicines with the potential to revolutionize the management of neurodegenerative and aging-related diseases, and in so doing, dramatically reduce the burden these illnesses place on patients, families, and societies worldwide. Our novel therapeutic approach targets a key regulator of metabolism and mitochondrial biogenesis, namely PGC1 alpha, that we have shown is downregulated in myeloid immune cells as a fundamental pathological process underlying a range of neurodegenerative and inflammatory diseases. And this forms the basis of the Tranquis platform, from which we have discovered and are developing a pipeline of small molecules. That pipeline is led by our clinical stage compound TQS168, which is positioned for ALS and other orphan neurodegenerative diseases. This innovation stems from groundbreaking research from the Stanford University Laboratory of Ed Engelman, which demonstrates that altered metabolism of myeloid immune cells underlies a variety of neurodegenerative diseases such as ALS, frontotemporal dementia, Parkinson's disease, and also has implications in Alzheimer's disease as well as other neuropathologies. We completed a $30 million Series A financing in June of 2020 with participation from SR1, Remiges, and Vivo Capital, and that has enabled us to complete phase 1 and clinical biomarker studies with TQS168, bring forward the next generation of compounds in preclinical development, as well as further progress the underlying platform. We have a strong IP estate with granted and pending composition of matter, use, and formulation patents. In addition to an experienced board, we benefit from a highly capable team covering the key disciplines needed for effective therapeutics, research and development, general administration, and entrepreneurial management. As mentioned, we are an immunoneurology company targeting PGC1 alpha to restore dysfunctional myeloid cells. PGC1 alpha is an important master regulator of cell energy metabolism. It is known that PGC1 alpha is downregulated in patients with neurodegenerative diseases. Myeloid immune cells, microglia in the brain and macrophages peripherally, are important for normal CNS physiology and homeostasis. Now, it's known that dysfunctional inflamed myeloid cells are a powerful, early, and continuous common pathway in multiple neurodegenerative diseases. Tranquis' scientific founders have demonstrated that PGC1 alpha is downregulated in myeloid cells in animal models of neurodegenerative diseases, and that upregulation of PGC1 alpha can restore normal myeloid cell function, with the potential to be a major breakthrough in the treatment of neurodegenerative diseases. From that discovery, we've developed a platform for discovering orally bioavailable small molecules that upregulate PGC1 alpha in myeloid cells with application to both neurodegenerative and non-CNS indications. That platform has yielded a pipeline of compounds at clinical, preclinical, and discovery stages of development, initially targeting orphan and prevalent neurodegenerative diseases. And it is this intersection of a dysfunctional metabolic pathway, an immune cell phenotype, and a major unmet area of clinical need that represents our core research strategy. As many of you may be aware, neuroinflammation is observed in multiple neurodegenerative diseases. For many years, conventional wisdom suggested that this observation was an epiphenomenon and not core to the underlying disease process. However, this understanding has been challenged recently, and it is now understood that myeloid immune cell dysfunction plays a critical and potentially causative role in many of these disease processes. We know that neurodegenerative disease begins with a specific injury or defect that results in deposition or accumulation of toxic material, such as iron, amyloid, or cholesterol, which in turn induces an inflammatory myeloid cell response. Myeloid cells normally perform a cleanup function in the brain, taking up and breaking down everything from dying or dead cells to foreign matter. However, if overwhelmed by these toxic stimuli, they become inflamed and secrete a range of tissue-damaging cytokines, thereby initiating and propagating a cycle of neurodegeneration. This is now recognized as being an early and powerful pathological pathway common to multiple neurodegenerative diseases. Our unique approach identifies myeloid immune cell dysfunction within and outside of the CNS to be at the core of this disease process. We identify and target master metabolic regulatory pathways that give us the ability to restore resolve the inflammatory state of these cells and significantly enhance their physiological function in the brain, for example, in microglia, and in the periphery, for example, in macrophages. Our approach has yielded drug candidates designed to not only stop myeloid cells from releasing inflammatory mediators, but also reprogram them to shift back from a pathological glycolytic state towards a healthy oxidative metabolic state, performing their normal physiological function, taking up and metabolizing the offending toxic stimuli to which I referred earlier. Taking this approach focused on an immune cell phenotype, we have designed, synthesized, and screened a series of compounds that comprise the Tranquis portfolio. Our lead compound, TQS168, is orally bioavailable, crosses the blood-brain barrier, and upregulates PGC1 alpha, restoring normal myeloid cell function with compelling effects in multiple disease models of ALS, frontotemporal dementia, Parkinson's disease, as well as some interesting early data in age-related neuropathologies such as Alzheimer's. Furthermore, we've identified an on-target mechanism-specific clinical biomarker of the effect of TQS168, which will accelerate its clinical development and reduce risk. Many of you are aware of the SOD1 model, which is a standard but challenging preclinical experiment for the detection of a potential therapeutic signal for ALS compound development. We have dosed TQS168 in multiple repeat SOD1 models of ALS at both Stanford and at Tranquis, where we demonstrate a highly significant improvement in both function and survival. On this slide, we show the results of the initial studies at Stanford, where we were able to demonstrate an extension of survival for 168 as compared to vehicle and against riluzole, which is an approved therapy for that indication. Furthermore, in the spinal cord staining on the far right, it shows a corresponding reduction in the presence of inflamed myeloid cells in the treated subjects. The early Stanford studies have been repeated at Tranquis with much larger sample sizes. And this slide shows the highly significant extension in survival from birth in the SOD1 mouse model, the median extension being 8 days. Interestingly, in this larger SOD1 study presented at last year's Society for Neuroscience conference, we were able to demonstrate a reduction in circulating inflammatory monocytes, which is the on-target biomarker for TQS168. Furthermore, when comparing inflammatory cytokines at day 120 as compared to day 90, TQS168 inhibited the increase of a series of key cytokines. Another example of the effect of TQS168 in the in vivo setting is in the TDP-43 model of ALS and frontotemporal dementia, where we have tested the compound initiating treatment after onset of disease. As for the SOD1 model, TQS168 demonstrates a dramatic improvement in survival in one of the most challenging but representative models of ALS and FTD. We show a similar effect in the transgenic alpha-synuclein model of Parkinson's disease, shown in the graph on the right. In BB2 cell microglia, the target cell type, we demonstrate a clear relationship between exposure to TQS168 and upregulation of the gene expressing PGC1 alpha. This is both a concentration and time-dependent response. The upregulation of the target gene corresponds with the reversal of the inflammatory phenotype in those myeloid immune cells, again shown here in BV2 cells. Here we show a concentration-dependent effect inhibiting TNF alpha secretion at nanomolar concentrations. We observe the same response, demonstrating the ability of TQS168 to suppress IL-6 secretion, again demonstrating nanomolar potency. So the proof of principle we've been able to establish preclinically is a very important feature of the Tranquis program. In essence, we've been able to demonstrate that TQS168 is able to upregulate the gene expressing PGC1 alpha, a master regulator of cell energy metabolism, in the target tissue, namely myeloid immune cells. In so doing, 168 demonstrates a significant improvement in survival and function in neurodegenerative disease models, such as the SOD1 ALS model, along with a corresponding reduction in the presence of inflamed myeloid cells in the CNS in these studies. Finally, we've shown that inflamed myeloid cells are increased in both frequency and activity in the blood of these ALS subjects. This biomarker, taken from blood samples from patients, is correspondingly resolved in a dose-dependent manner when incubated ex vivo with TQS168. So with our technology, we are able to demonstrate engagement with a biomarker from patients specific to the TQS168 mechanism of action, establishing target engagement, and that engagement corresponds with improved survival and function in established animal models of disease. Our overall R&D strategy is focused on three key drivers of growth and value. We've recently initiated phase 1 with our lead compound, TQS168. This compound is positioned for orphan neurodegenerative diseases, ALS being the first indication. Proof of concept in that indication will not only enable 168 to move rapidly into pivotal studies for ALS, but also positions the compound for exploratory studies in other orphan neurodegenerative disease indications, where this combined immunometabolic mechanism has the potential to offer a major therapeutic advance. In addition to the opportunity presented by 168 as a standalone asset, it also serves to prove in early clinical development the overall concept of targeting PGC1 alpha in myeloid cells through the associated use of a mechanism-specific peripheral biomarker. Clinical demonstration of our ability to be able to fundamentally reprogram myeloid immune cells, reverse inflammation and the pathological phenotype, and restore normal function will be of huge value for the validation of our upstream pipeline compounds and the underlying discovery platform. In addition to 168, we have a follow-up compound in development, namely TQS621, which targets the same mechanism, but is positioned for broader indications such as Parkinson's and Alzheimer's. Last year, we initiated a campaign of design, synthesis, and screening of a next generation of compounds targeting this mechanism. This has yielded a series of hits from which we anticipate generating lead compounds for development during the course of this year. So the execution of the aforementioned strategy over the next 2 years will result in us moving from early clinical stage development today to being in mid to late stage clinical development with TQS168, in early clinical studies with TQS621, and with 2 to 4 next generation compounds of various stages of development from lead optimization through phase 1. During the next 18 to 24 months, we anticipate hitting multiple inflection points, beginning with the initiation of healthy volunteer and biomarker studies with TQS168, with a move into ALS patients during the second half of this year, as well as completing preclinical development with TQS621 and nominating next generation leads. So in summary, Tranquis Therapeutics is discovering and developing innovative medicines with the potential to revolutionize the management of neurodegenerative and aging-related diseases. We are using our strengths and history in myeloid cell biology to identify the role of fundamental regulators of metabolism in those cells in health and disease in order to discover novel therapeutics that can reprogram dysfunctional myeloid cells and restore them to normal function. This approach underlies our platform from which we have discovered and are developing a pipeline of orally bioavailable brain-penetrant small molecules, initially targeting neurodegenerative diseases, and these are now at clinical, preclinical, and discovery stages of development. I thank you for your attention and encourage you to follow up with us via the biopartnering site if you have any questions. Thank you very much for your attention.